Thank you very much, Professor Ramsey. We will immediately continue with the next speaker, and then we'll do jointly questions. So also people online can put questions for both speakers, and we will answer them at the end. So the next speaker is Dr. Charles Rotimi. He's director of the intramural unit of the National Human Genome Research Initiative, studying genomics and global health. He's been instrumental to the launch of the E03 Africa study, acted in the National Academy of Medicine in 2018. He is founder of the African Society of Human Genetics, and he is the 2023 president of the American Society of Human Genetics. It's really fantastic to have you here with us. He investigates genetics and health disparities. He is also, important again, co-chair of the conference organizing committee for the ECHG 2023, and will talk to us about diversity in genomic medicine. Charles, the floor is yours.
Thank you very much. Thanks for that introduction. And really glad to be here in person and for us to begin to reconnect after our 3 years of COVID. So it's really good to see everybody. And I'm not quite sure what to do with my mask now, whether to put it on or put it down. And so I think all of us, we are dealing with these different challenges, in our own way. So again, welcome, and really wonderful to interact in person. My talk today is really trying to see how diversity at the global level, not just in Africa, is important for how we deploy genomics to different parts of the world as a scientific imperative and also as a social justice issue. Some of the examples you saw in Michelle giving a very comprehensive presentation also will play in mind, but hopefully I can give it a different perspective, in that regard. So, like I said, I think increasing diversity is a scientific imperative, and it's a missed opportunity to make discoveries and to understand human history. And I hope to provide you with examples of that in my talk. So what do we mean by lack of diversity? I want to focus your attention here, again, in terms of the participation from different parts of the world. You will see that it's absolutely not even. And one of the things that I also try to show here is why is this playing out, and why is it very difficult for us to begin to create equity across this genomics in different world populations. So hopefully I can provide some insights into those kind of questions, and maybe leave sufficient time for us to have discussions again and ask different questions. So as you heard from Michelle, there are multiple reasons. One of the things I learned from the days of HapMap and 1,000 genome and up to today is that the global distribution of human genetic variation is not random. They have geographical structuring and all the things that impact individuals, families, and ancestry and geography are critically important as we understand human genetic variations. And we have different factors that have shaped our genome and left different signatures. So for me, the way I look at our genome, I look at it as a history book that we are just beginning to learn how to read and to interpret. And I want to challenge all of us to do this reading and understanding using new technology, to do it in such a way that we don't reaffirm old prejudice or old way of looking at our data. We should allow the data to guide us and to see how we are related and also how we are different across the world. So again, I'm just going to go through this very quickly because Michelle reviewed this. But the point I want to really highlight is the number 4 there, which shows that we are indeed very similar. And the reason for that is that, again, most of us, or if not all of us, started our history somewhere in Africa. Hence, we share a lot of our genetic ancestry. But some of us left because as humans, we like to travel. And when we do travel and go to different parts of the world, we interact and we share our DNA. And then we continue to weave these very complex structures of our genome. But there are parts of our genome that you can only study in African ancestry populations just because the original people who migrated only took a subset of the variation that existed at that point. So you saw this, I think Michelle also showed this, but the point I want to make in this particular slide is that, again, variation is not random, it's structured, and we need to study it. And in terms of the African continent, whether it's SNP or non-synonymous or loss of function variation, you do see that African genomes tend to display more variability. And it's the same thing in terms of haplotype. So what I want to do in the next slide is really to use this diversity in haplotype structure in terms of fine mapping in a study that we published recently in my lab. I hope this video will play. Trying to see.
In this manuscript, Dr. Charles Rotimi's group at the National Human Genome Research Institute identified the genetic architecture of circulating resistant levels in Africans from Nigeria, Kenya, and Ghana, and African Americans from the Washington, D.C. area. Resistin is a small peptide secreted by bone marrow cells in humans, and it's been suggested as a biomarker for metabolic syndrome and type 2 diabetes. 2 previous genome-wide association studies performed in people of European and Asian descent identified significant associations with genetic variants near the RETN gene, that's the gene that encodes resistin. Dr. Rotimi's group performed GWAS for plasma-resistant levels using more than 18 million genetic markers in nearly 4,000 continental Africans and nearly 2,000 African Americans separately. Both GWAS identified the same variant, 75 kilobases upstream of the RETN gene, as the most significantly associated SNP. The haplotype structure in the African populations allowed the researchers to fine map this locus to a single variant, and whole exome sequencing studies ruled out the contributions of coding variant in the RETN gene. Further, the GTEx dataset demonstrated EQTL effect of the index variant on resistin expression in immune cells. Together, these results suggest that regulatory variants that affect resistin expression levels derive the variation in plasma resistin levels. Finally, the authors performed Mendelian randomization analysis of resistin levels on metabolic phenotypes. However, they did not find evidence of a causal association between resistin levels and metabolic syndrome-related phenotypes. Overall, this well-executed study was able to fine map the RETN locus for resistin associations and, perhaps more interestingly, cast some doubts on the idea of resistin as a biomarker for type 2 diabetes.
Thank you. I hope you enjoyed that animation, which I love, and I try to use it in any talk I give these days. So fantastic work. I commend the journal, actually, for doing that. Again, this is a work from my lab at the NIH. The point here is really that despite a huge amount of very productive discovery in European ancestry populations, we are indeed still making discoveries in African populations. This is a study we've been doing for the past 20 or so years called Africa-America Diabetes Mellitus Study, ADAM, with sites in Kenya, Nigeria, and Ghana. We're trying to understand the genetics of type 2 diabetes. So what did we do? Find in about 6,000 or so individuals, we made a novel discovery in a gene called ZRAMB3. The variant that led us to this gene is, I want to use the concept of African ancestry specific. I'm a little bit reluctant to use things like African specific in the sense that it creates the impression that all Africans have it, and that is not true. But here, just again for simplicity, I'm saying African ancestry specific because when we look in published databases, it's indeed rare, but it's not a characteristic of being African. I think that's really the major difference here. So what does ZRAMB3 do? When we made this discovery, we really didn't know because we looked at the literature and we didn't really see any major insight into ZRAMB3 and how it may impact diabetes risk and glucose metabolism. So we went to a model organism, zebrafish, and by knocking down ZRAMB3 in zebrafish, we were able to demonstrate a radically reduced number of beta cell and beta cell mass, which was the first insight that we gained in terms of how ZRMB3 may indeed impact blood sugar and also diabetes risk. So we further took this discovery to mammalian cell lines, and what we did here was to use a small molecular RNA and to silence ZRMB3. And what you will see here is among the controls, there's really a normal response to a high glucose environment when the ZRMB3 is not disturbed. But when you silence ZRMB3, you will see that in this high glucose environment, those cells have very diminished insulin response, and therefore beginning to give us insight as to how this may indeed be important in type 2 diabetes. One of the things that we noticed a few years ago in our study of diabetes in West Africa is that we do see individuals who are type 2, but very early in their course are insulin requiring. And we are thinking that this may give us insight into why that is playing out. So we are moving to the next phase of this study, and our goal is really to stratify individuals using this variant and see whether we can indeed provide additional insight into how and why some people may require insulin early in their course. Because one of the things that physicians tend to do is they wait and they use oral treatment first, or even a lifestyle modification. But for some of these individuals who require insulin, if we are able to detect them early enough, we may be able to make interventions much earlier in the process before organ damage starts. So the other example here in terms of diversity is really, again, I'm going to try to go through very quickly because I think Michelle made a very great point in all of this. Really, the opportunity to make novel discoveries. This is a study that, again, I was a co-author on, but it's a huge study, I think over 300 authors, about 1.6 million individuals, and with about 350,000 non-European individuals. But the point here is really when you look at discovery, you will see that individuals of African ancestry, we have ability to make much more detailed and novel discoveries in these very populations. And the same thing here in terms of proportional variance identified from ancestry-specific meta-analysis that is powered enough to see the same effect, but with ancestry-specific frequencies. The point I want to make here is in this dark blue region here. So what you do see is that it's likely to, for all those sample discoveries, when you look at African ancestry populations, you do find that the ability to have the same kind of power is really diminished, again, because of the variability that you see in African ancestry populations here. So again, giving us insight into how and why we need to do more discoveries. The other point is Neanderthal. This, to me, is absolutely fascinating story. I read just about all the Neanderthal publications that come out because I just feel that there was no way for us to gain this kind of insight without genomics. To me, this is one of the great demonstrations of the ability to use genomics to understand human history and also to understand human disease. So, like I said, we humans, when we migrate, we share DNA. Sometimes we even share DNA with species that may not necessarily be a part of our initial inheritance out of Africa. And that is why I think so far the data seems to suggest that most of the introgression in terms of the Neanderthal is occurred outside of Africa, but I think the jury is still out on that. And that introgression seems to impact the genome of people in Europe and Asia, to about 4% or so. And the question is, does this part of the genome have importance for human traits and diseases? And this is a wonderful illustration of how the introgression from Neanderthal actually may indeed be very important in understanding human diseases. This is a type 2 diabetes study that was done in a little over 8,000 Mexicans and other Latin American populations by the Sigma Type 2 Diabetes Consortium. And that was a major discovery in terms of the solo carrier and having a very specific haplotype that you see here. But what was interesting in this haplotype is that it is more common in people with Native American ancestry. But when the Asian genome, archaic genome, was compared, in terms of trying to understand where did this variant come from, it became quite clear that the sequence, this high-risk haplotype introgression into modern humans actually came as a result of Neanderthal admixture. Again, giving us that this part of the genome that came from Neanderthal may indeed have biological function, and that we need to pay attention to it and study it. And I think I have another example. Again, this is using COVID. This paper was published in New England Journal of Medicine in 2020. Where they made this discovery here, I want to pay attention to chromosome 3 that had a very high significance with COVID risk. And it was later also traced to the Neanderthal part of the human genome. And the frequency of this particular variant is indeed much higher when you look at South Asians and also in Europeans. And there's speculation that this higher frequency of this genomic region in persons of Bangladeshi's ancestry may help explain their 2 times mortality from COVID compared to the general UK population. So again, beginning to tell us why it's important for us to study different human populations, and perhaps also to pay attention to how the introgressions, that part of our genome, is indeed important in biology. So again, looking at Asian population, I absolutely love this study when it came out because it helps me to make the point about the importance of diversity. Again, this is a 100,000 project from across Asia, which demonstrated that the current databases is indeed not very, very efficient or comprehensive enough to truly understand the genetics across Asian populations, whether it's in form of novel cancer, understanding and variants, and also information about enrichment for something like beta thalassemia variants, which is again up to 1.4% in South Asia. You can see how that enrichment can help us to understand the genetics of beta thalassemia quite more efficiently. And H3 Africa, again, I will not complete this talk without talking about H3 Africa. To use this as an example of how we are trying to close the gap in terms of equity in genomics, especially in the context of Africa. To me, this is a project that is absolutely dear to my heart, and we have been doing this now for 10 years. We just celebrated, like Michelle said, in Abuja, Nigeria, 10 years of continuous funding for this research. And what we are hoping to do with H3 Africa is also to create a global good. We are going to make sure that this data is available to the world population to use and to understand the genetics of African people, but perhaps just as importantly, to understand the genetics of the global population. Africa is our home, and there are things that we can see there that we may not be able to see in other parts of the world, but it doesn't mean that understanding those biology will not be relevant to the other parts of the world. So I think it's really important for us to appreciate these dynamics. Now, Michelle showed some of this data, but the point I want to make here is the last bullet, new insight into pathogenic and clinically relevant variants. when we published this study, to me, it was absolutely mind-blowing that there are variants that are being classified as pathogenic when you look at something like, ClinVal and other, databases. But a lot of Africans have these variants, and they are fine, and they are moving around in their community, which brings to question, are those variants actually pathogenic? And if they are indeed pathogenic in a certain environment compared to others, then it forces us to ask the question, what are the modifying background for these variants to be pathogenic in certain environment and not in others? Again, showing the importance of doing diversity work. I'm not going to go through this because Michelle showed you this data, but the point here is for this particular observation is that we, as Africans, analyze this data, and we absolutely surprise ourselves about our own lack of understanding of migration across Africa. And so the question here is, how do we systematically sample Africa to truly understand the migrational pattern that has occurred across Africa for so many, many years? And I do not have the right answer to that, but I think it's something that we really need to think about very carefully. Because whenever we sample a new African population, we make novel discoveries. And as Michelle also showed, we are nowhere near the plateauing in terms of variant discovery or understanding human history. But the question remains, and this is where I want to challenge the population geneticists in the audience, is to how do you model a geographical area like Africa that has evolved for so many years? How do you begin to model that to see how many of these African populations that you can truly sample to begin to describe the diversity in a more comprehensive manner? To me, I think that's a challenge for all of us in human genetics. Now, I started earlier by saying lack of diversity is a scientific imperative, and I also said it's a social justice issue. The question is, as we use genomics to develop new therapeutics, are we going to deploy that in a way that it will be beneficial to all world population? I think that's the question we have to constantly ask ourselves. Are we training the right workforce? Are we even training the right genetic counselors to help us to deploy genomics in the way that we want to do it? And this study here that was published in the England Journal of Medicine, but highlighted by New York Times, actually showed how poor reference databases can lead to misdiagnosis. In this case, for individuals called blacks within the United States, where there was discovery or the speculation that there was heart disease among these individuals, but it was actually due to the fact that the reference database did not have enough representation of individuals of African ancestry. So, polygenic risk scores. This is, I think, our first comprehensive attempt to begin to get the fruit of GWAS in terms of clinical practice. And I'm afraid that if we don't do this very well, this may represent our very first example of exacerbating health inequalities around the world. Because of our emphasis, again, I'm not blaming scientists, I'm just saying that's the reality of what we have been doing. We've been doing most of this in European ancestry populations because that's where the large cohorts were, that's where the funding was. And again, I'm very, very careful not to use that as if it's a blame. It's not a blame at all, it's just the reality of where we are. But the other reality of where we are is that when we use European ancestry discoveries, we do notice that our prediction capability decreases quite enormously as you move from Europe to different parts of the world, in a sense as a principal component in terms of distance from the UK. So the question now becomes, how do we actually deploy this in a place like Nigeria or Kenya, and without necessarily making false recommendations to individuals? I think it's really a major challenge that we need to pay attention to. And there is some hope that if we indeed increase diversity, that we don't necessarily have to create different predictive cohorts, that if we have sufficient number of diversity within a specific cohort, in this particular study of about 1.6 million individuals, that we can indeed develop trans-ancestry polygenic risk scores that may help us to be more predictive in different human populations. And the example here, if you look at the blue color, that is the trans-ancestry polygenic risk scores. You will see that as you go across all of these populations, that the polygenic risk scores from trans-ancestry actually is quite predictive as you go across the spectrum, which is, again, a little bit of comfort, but it's still being driven by the fact that we have multiple ancestries in terms of that construction of polygenic risk scores. So, but I want to caution us that while trans-ancestry polygenic risk scores may be very important for some traits, it may not work for other traits. And I think the example here is, is type 2 diabetes. That for lipids, it tends to be working quite well, but for type 2 diabetes, it seems to be not as predictive for reasons that we need to adequately understand, so that in some instances, we may have to do population-specific discoveries or ancestry-specific discoveries to be able to truly deploy polygenic risk scores. Now, I want to talk in the context of gene-environment interaction. All my career, I have been interested in understanding health disparity and how our environment and our genome shape differential susceptibility to disease and also differential response to therapeutics and things like that. So I have always been very curious. I always caution my lab. I say we are a genomic lab, but we cannot forget the environment, especially in the context of health disparity. And I just wanted to, again, show an example using the APOL1 variant. Again, for me, when this study was published, it was absolutely wonderful. But that if you carry 2 copies of these renal risk variants, that your odd ratio is over 10. What was interesting, again, as human geneticists, is that when you have a variant that is this deleterious, the evolutionary process tends to eliminate it. And therefore, the question becomes, why did it rise to such high level in West Africa, especially, and some parts of East Africa? So the frequency is about 40% among the Yoruba. And again, you find the same sort of frequency in Ghana and other West African populations. It's about 20% in African Americans, which you would expect as a result of admixture. The African American gene pool is about 80% European and about 20% West African. So this is absolutely consistent in terms of frequency as it relates to the APOL1 variant. It turned out, after further experimentation, that the APOL1 risk variant, kidney risk variant, is actually protective against the so-called African sleeping sickness, infections in terms of trypanosomiasis, because it lyses trypanosomes. Again, so you can see in the evolutionary context, you will die faster from the infectious process in terms of African sickness before you develop kidney disease. So it makes sense that this protection rose to such high frequency. But the thing here is that in an environment in the US or other Black population around the world, where you don't necessarily need to be protected from African sleeping sickness, this variant is absolutely devastating to kidney disease. So just as a little epidemiological data, African Americans make up 13% of the US population, but they make up 32% of kidney failures in the US. So you can see tremendous impact. And African Americans are 4 times more likely to develop kidney failure than their white counterpart in the United States. So this has been a hair scratcher for everybody for a very, very long time. Why is this? So there are speculations that it could be hypertension, and other morbid conditions. But it turns out that 1 in 5 African Americans with 2 copies of the APOL1 will develop kidney disease. And the APOL1 variant accounts for 70% of non-diabetic kidney failure in African Americans. And about 4 in 10 African Americans who are on dialysis is due to having 2 copies of the APOL1 variant. So you can see how this is already helping us to understand some of that disparity that we see in terms of kidney failure. Now, it already has very serious implication. There is no way to use skin color to know who is going to carry this variant because of admixture, and it has spread all over the world. But the origin, again, is as a result of protection from trypanosomal infection in Western and some parts of East Africa. But it also has a very interesting clinical importance already, even while we are trying to develop the drugs to help treat this condition. If you are a kidney donor, you want to make sure that you understand your APOL1 risk, whether you carry 2 copies of this variant, because the likelihood of the remaining kidney to fail is extremely high. And also, if you're a kidney recipient, you want to know the status of your donor, because if they have 2 copies, the likelihood of that kidney that you are receiving, the likelihood of it failing is also quite high. So you can see that it already has implications for understanding the clinical care and transplantations in terms of kidney organs. So this is an example, again published by Amy Bentley from my lab as part of the CHARGE consortium, where we try to understand gene-environment interaction in the context of lipids and smoking. Now, when we published this, when we analyzed this data, we discovered that most of the variants that we picked up as a result of this interaction were indeed African ancestry specific, or in the trans ancestry analysis that also included African ancestry individuals. And our initial thinking was, oh, this must be due to allele frequency differences. But it turned out that that is not really the case. That for this example, this specific SNP on this right side of this cartoon, you will see that, where's my cursor? That the allele frequency is actually higher in the non-African ancestry populations compared to the African ancestry populations. But you'll see that there's absolutely no association in the non-African ancestry populations, whereas in all of the African ancestry populations, you do see quite significant associations. And this particular SNP, again, is an EQTL for a taste receptor that affects cigarette smoking. And a group from Hopkins and NIH, a separate group, not my lab, made this very interesting discovery of a haplotype that increases 5 to 8-fold the odds of menthol cigarette smoking. One of the things that we've known for years is that African Americans who smoke, 80% of them prefer menthol cigarettes. For a very long time, we did not know why that was the case. Now, that has a very significant implication because menthol cigarettes, menthol itself, it creates a smoothing effect when you smoke. And therefore, you tend to inhale more, and also you tend to pull in more of the carcinogens. And so discovering this haplotype that may indeed increase the odds of menthol cigarette smoking, and also that it is more common in African ancestry individuals, to me was absolutely an interesting discovery. And it's also part of the movement right now, I don't know if you've been following, that the FDA, which is the Food and Drug Administration in the United States, there's a real strong movement now to ban all menthol cigarettes. again, as a result of some of this discovery and understanding. So, this is one of the prototypical examples of gene-environment interactions that I have ever seen. And that is adaptive capability to a toxin, in this particular case, arsenic. And if this doesn't convince you about why we need to do genetics across the global population, I don't think anything else will. So the groundwater of this community in Argentina is contaminated by arsenic, over 20 times acceptable levels. It turns out that the local population may have indeed developed adaptive capability to a very dangerous chemical, arsenic. What has happened is that for arsenic to be less toxic, you have to metabolize it from the MMA to the DMA. And this population has indeed developed the capability to do just that. And therefore, they are protected against the very dangerous impact of arsenic in their drinking water. What actually was very interesting in this particular instance is the fact that even the very close community around this village actually displayed lower frequency of this particular haplotype, which absolutely points to the fact that it is due to positive selection as an adaptive capability to arsenic in their water. So to me, it really does demonstrate the first example of human adaptation to a toxic environment. And I wonder how many of such are there as we study global populations. So I think I have a couple more slides and I will end my talk, but I wanted to really share this slide. This is a work again from my lab where we studied about 6,000 individuals across the world from 30 primary language families. And this cartoon here shows where the samples came from. And what we discover when we use a clustering algorithm, which basically we ignore all of the ancestry information that was provided to us, and basically asked, cluster this huge database and let us know how many ancestries we can discover across the world. For this particular data set, we observed 21 ancestries globally. And again, to emphasize the diversity across Africa, 11 of these 21 was in Africa alone. Now, ladies and gentlemen, this is the message I want all of us to take home, because I think if we tell this story more, I think we will love ourselves and we will appreciate ourselves more as indeed a mosaic of complex ancestries. 97.3% of individuals in this data set had mixed ancestry, of average of 4 ancestry per individuals. And there was no part of the world where we didn't see tremendous admixture of different populations. And the caution here is that due to ubiquity of ancestry, we need to account for this in how we deploy genomics, how we do our work, and even how we interpret our data. And I want to use an example, a real-life example here, to say that by not acknowledging the complexity of ancestry, we can indeed make very bad clinical decisions and also public health decisions. This is a study, again, that was published a few years ago, where it displays typically when somebody appears before a physician, especially in a place like the United States, and I'm sure it's in other parts of the world, the first thing the physician will notice is, okay, Charles, this is a black man, and then you will say, oh, how old are you? So you see how the characteristic starts, and those characteristics come with certain assumptions. In this particular case, this is a serious negative consequences of a physician's assumption about a patient's race. An 8-year-old boy, phenotypically European, presented with acute abdominal pain and anemia with a hematocrit of 0.21. Said although his body temperature was only 37.9, surgery was considered until a technician found a red coupled with hemolytic characteristics on the smear. Surgery was subsequently cancelled, but what is important here is that this kid, because it's European phenotypically, has never been diagnosed of having sickle cell anemia, and this kid actually has sickle cell anemia. Now, if this physician or physicians that have seen this kid over the years were able to go beyond his phenotypic appearance to actually ask questions about his ancestry, then you will understand where this has come from, because his parents were from Grenada and were of Indian, Northern European, and Mediterranean ancestry. So if you know the sickle cell story, you will know that this should not be a surprise discovery, but it wasn't done, again, because of the way we look at each other. And we need to find a way to go beyond phenotypic appearances, especially as we deploy genomics, because there is no way for you to really know what I carry or what my mother carries. And the other example I want to use is the story I always tell about myself and my mother. I grew up in Nigeria. Some years ago, I usually would say 18 plus X, and you have to determine what X is. But I used to get malaria like 2-3 times a year. And then, this also tells you about my age, the only medication that was really effective then was chloroquine. And when we take chloroquine, it does fantastic, it cures the malaria. But myself and my mother have very different reactions to chloroquine. I will itch for days, I can't sleep, but my mother will just be fine. So I always say that if you wanted to understand the pharmacogenetics of response to chloroquine, my mother and myself would belong to a different group. So the point here is that we have to go beyond how we look. Because I look like this, I cannot represent all people that look like me. Again, I think that is really important for us as we deploy genomics. So I'm going to conclude by saying the lack of diversity hinders our understanding of biology, exacerbates health inequalities, raises the question whether genomic-derived therapeutics will serve all human populations equally. Diversity in genomics continues to be disproportionate, and we are making progress, but it's really difficult. There has to be a way to make funding available to actually increase global cohorts so that we can indeed begin to approach equity. And findings from European may not broadly apply to all other populations because of ethnic-specific variants, and also inter-ethnic differences in allele frequencies and all other consequences. But perhaps for those who may not be too keen on the social justice issue, may be keen on the fact that it is also a scientific imperative. We will not truly understand genomics if we do not deploy it across the world and study different human populations. Like I said, we are making progress. These are examples of studies that are beginning to address diversity across genomics. It's a work in progress, but it's really, really fantastic. I want to particularly point out TOBMED, which was done by the Lung and Blood Institute at NIH, which has sequenced, I think, over 160,000 individuals now, and a large proportion of that is non-European ancestry populations. So if you are interested in using those kind of data set, I think TOBMED will be a very good resource to be aware of. And again, I want to thank you, and I'm glad to take questions. And this is me, one of our H3 Africa studies. I think this was in Zambia. And this is me pretending to be a warrior that I know nothing about. Thank you very much.
Thank you very much. Charles, if you come sit here, and Michelle, can you also join us? I will move, and we open up for questions. For discussion. So, we have more than 5 minutes for questions. Please introduce yourself and ask a question.
Hi, my name is Caroline Kuchenbecker from UCL in the United Kingdom. Thank you both for the fantastic talks. That was incredibly interesting. I have a lot of questions, but I'm prioritizing the one about where do you see the research priorities in terms of genetics, both on the African continent and maybe for diverse populations in other countries, what are we not doing right now, and what should we be studying in terms of conditions, groups, etc.?
I think for me, one of the most important things that we have to do, first of all, is to recognize the current limitations of existing databases. The fact that we just have not done enough work across different populations, and therefore funding agencies needs to provide focused funding, whether it's South America, whether it's some parts of Asia, or some parts of Africa. We need to find a way to fund different cohorts so that we can indeed understand all the signatures in our genomes are due to different geographical restricted exposures. The other thing also is that there are rare conditions. They are not necessarily rare in terms of typical understanding of rare conditions. They are indeed of importance to various local environments. For example, there's a condition that we have been studying in Ethiopia called podoconiosis, for example. Is due to exposure to fine-grained soil. And it's local to that environment. It has been in other parts of the world, but because people wear shoes now, it's less common. So if we don't study different populations, we may not be able to use genomics to understand such conditions. So there is a need to also understand different disease distributions across the world. I hope that provides some response to the question.
I think that's a great response, Charles. I think, we really need to study longitudinal data in understudied populations. I think we don't have enough population cross-sectional studies, and that we really need to collect that complex data in populations to look at common conditions, which we so poorly understand, but contribute so much to the burden of disease. So, I know you have an interest in mental health, and that is so important to study. We really don't know enough from non-European populations, and there will be new things to discover from these understudied populations.
Okay, thank you very much. Next question. I think it should be fine. Can you speak, right?
Hello? No, it's not. No.
Yes.
Okay. Okay, so I have 3 questions actually. One of them is, what about the people of the North Africa? Like, is it expected that they would be of more diversity, higher diversity, being coming from the Arabian Peninsula and admixed with Europeans as well? This is the first question. My second question is how to access some of the African data, like regarding genomics or other metabolic phenotypes, in order to compare or to do meta-analysis. And the third question was about type 2 diabetes, GWAS, how many samples were involved, and what is the prevalence of type 2 diabetes in that population?
I'll take your last question, and maybe Michelle can deal with the others. The last question is our type 2 diabetes project. Right now, we have about 7,000 individuals, both cases and controls, enrolled in that study. But for that particular GWAS publication, I think we used roughly about 6,000 individuals because it's still ongoing. And if you are interested in collaborating on that kind of project, please don't hesitate at all. The reason why it's currently not, part of it is not on the public database, is that when we started our study about 20 years ago, our informed consent was not broad enough for us to share some aspect of that data, so we can only use it for collaboration. But the new part of the study, which is roughly now about 2,000 individuals, we are going to put all of that on the public database. So anybody who wants to interrogate that study can indeed use it for the work that they do. Again, I think for all of us, it is critically important that we share data. That is truly how we can indeed make impact across the world. There's no one laboratory that can make all the discoveries. So I absolutely believe in data sharing, and I think we have to do that at a global level.
Thanks, Charles, and I'm happy to briefly address your first 2 questions. So the one about North Africa, I think we don't know nearly enough about the population genetics of North Africa, extremely complex, multiple admixtures, and I'm very happy to say that we're going to do a project now on 1,000 African genomes, of which 200 to 300 will be from North Africa. So I'm hoping that we'll get more insights there.
Anything from Egypt?
Say that again?
Anything from Egypt? From Egypt.
Interestingly, we're looking at Tunisia, Mauritania, and sure, can't remember what the third one is, but you're right that Egypt is also understudied. So I think there are genome projects that are starting now in northern African regions. So the second question, in terms of the H3 Africa data, most of that data is in EGA, so you can get both phenotype and genomic data, and it's under managed access, so there is a data and biospecimen access committee which will give permissions for studying that data. But again, as Charles said, the intent is to share it widely, because I think globally we'll get more insights if more people study the data.
So currently we can
I'm sorry. We have to actually move on. I want to have very quickly one online question, and then a very quickly question there, and then we finish. So I had a question from Hilde Schrempf, who asks whether you have compared genetic variation in genes involved in basically in nutrition, so in digestion food, whether you have any things there that are different among populations.
Yeah, we did a study a few years ago where we looked at nutrition, how nutrition shapes our genome, it is very clear, for example, that across global population, not just in Africa, that populations that have had to digest starch have multiple copies of the amylase gene, and that is what is needed for digestion of starch. So there is definitely dietary structuring of the distribution of that particular copies of that particular gene across global populations. Yeah.
Okay, and then final question. Yeah, sorry.
No, not a problem at all.
No, actually there's a person behind you. Sorry. I have to make a choice, so perhaps you were there before. Go ahead.
I saw that you spoke about using other biomarkers. I think I saw most of them were lipid biomarkers. Are there any other biomarkers that you were looking at in combination with your genomics work from other omics?
Oh, yeah. Definitely. Part of the diabetes project that I displayed, we are right now in the field getting biopsy samples in terms of muscle and adipose tissue. Again, that is really to help us in terms of expression profiling and also to do single cell, type analysis. And the goal is really to use multi-omics. We are also collecting stool samples to do microbiome and proteomics. So we are trying to combine all of this strategy to truly understand how all of these things come together to give us insight into not just discovery of variants, but to understand what those variants actually do and how they impact health. Yeah.
Don't forget the environment, right?
All right. I want to thank both speakers for a fantastic session, and I hope you will all join us in Cape Town in February next year. Thank you very, very much.